The next chapter we're going to take a look at is Chapter 7, which discusses the generation of humoral effector mechanisms. So essentially what we're going to be looking at is the B cell response. What you need to focus on specifically are the humoral responses to thymus independent and dependent antigens, and we'll go through that. What are the surface molecules that contribute to the production of the B and T cell conjugates, the basic structures of all of the 5 antibody isotypes and their function, and that's really high yield for step 1. We're going to talk about some definitions, the meaning of valence, affinity, and avidity, also the role of somatic hypermutation and affinity maturation during the immune response. We are going to look in this chapter at complement. I know it's everybody's favorite topic, but we have some good news on the complement front that we'll talk about when we get there. Most important for complement is what are the biological functions of the complement component. So really, that's what we're going to focus on when we get to the complement cascade. Now, most of you know when we're talking about humoral immunity, we're talking about antibodies specifically. So we're looking at your immune response's defense against things that are extracellular. And predominantly, really, we're talking here about bacteria, extracellular bacteria, like staph and strep, for example. Now, B cells, in order to be really effective, really need the help of T cells. Remember, in our grand scheme of immunity, you have your T helper cell, and remember, they're the boss. They're the boss over all of the other cells. So here, what they're doing is they're going to communicate with the B cell so that the B cell knows how to make really good antibody molecules that have a wide variety of biological effector functions. So as you look through this diagram, we're just going to focus on the interaction between the B cell and the T cell. First, you can just see that the B cell is functioning just like an antigen presenting cell. So it's going to process antigen, although differently than a macrophage or a dendritic cell, because a B cell is going to do it specifically, because the antigen is going to specifically now bind to the B cell receptor, which, remember, is a membrane-bound antibody molecule. So as the antigen comes in, the B cell processes it and presents it on the surface of a class 2 MHC to a T helper cell. So now that T helper cell and a T cell receptor can bind to the antigen and class 2 MHC. That's the first thing that needs to happen. Now, another molecule that's important in this interaction is going to be the interaction between CD28 and B7. CD28 is the molecule that's on the T cell, B7 is on antigen presenting cells, and that's really what the T cell needs to become activated, okay? What the B cell is looking for from the T cell is the CD40 ligand. So the third interaction that we're looking at here is we're looking at the interaction between CD40 and CD40 ligand. That is an essential interaction. That's one you definitely need to start and you need to know, because there are a bunch of effects that won't occur if CD40 and CD40 ligand don't interact. Class switching is definitely one of them. So you need that CD40 ligand interaction for class switching. There is an immunodeficiency disease that's associated with failure of that interaction, which is called hyper-IgM syndrome, which we'll talk about in a little bit. But you also don't get a memory response if CD40 and CD40 ligand don't interact. So that's important. Once you have all those steps come together, the T cell is now going to help the B cell out by way of class switching. So the T cell gives the B cell some cytokines, depending on the cytokines that the T cell gives the B cell, and you can see a list of different cytokines here. Depending on the cytokines the B cell receives from the T cell, it will know to class switch from IgM into either IgA, IgG, or IgE. Okay, so that is how that conjugate forms. We're now going to look at a little bit of an animation just to show you a little bit of the temporal regulation of how that occurs. Again, T cell interacting with a B cell. The B cell is functioning as an antigen-presenting cell. So the first thing it has to do is bind antigen, which it does, but specifically through the B cell receptor. Now it will internalize that, process it, and present it through that exogenous pathway. And so now it will be presented on the surface of a class II MHC molecule. Now, the B cell and the T cell come together, and you can see all of those molecules we talked about. The first between the T cell receptor and the class II MHC and antigen. The second we talked about was the CD28-B7 interaction, and then the other interaction was CD40-CD40 ligand. Now, the other set of cell adhesion markers you see is between CD2 and LFA3. Now, think about what's going on with the cytokines. The T cell is giving the B cell cytokines, and in order for the B cell to really have a chance to get those cytokines, those cells have to be tethered together for a good period of time. The B7-CD28 interaction, the CD40-CD40 ligand interaction isn't strong enough to hold those cells together long enough. So that's what the CD2-LFA3 interaction. It's simply a tethering molecule to keep the cells together long enough for cytokines to go from one cell to another. Now, once that occurs, you're now going to have the cytokines, and again, depending on the response, depending on which antibody is needed, you're going to have anywhere from the IL-4, IL-5, and some of the other cytokines that now allow the B cell to undergo class switching. So now again, we're talking about switching from IgM, which all B cells start out making, IgM, and now we're going to switch to something that's a little bit more effective in the immune response. Now, take a step back. Without the help of T cells, what can a B cell do? Can a B cell do anything without the help of T cells? It can. It's just not, it's not the best response. In the case of a thymus-independent antigen, in this case, it probably wouldn't be a peptide, so things like polysaccharides. And the best example is LPS on the surface of gram-negative bacteria is a thymus-independent antigen. When you don't have the help of T cells, the only antibody you can ever make is IgM. Now, why is that? Remember, I said earlier, CD40, CD40 ligand have to come together in order to get class switching. If there's no T cells, there's no CD40 ligand. So the only antibody you can ever get is IgM. You do not get a memory response like we talked about in a little bit. And mostly, again, we're talking about large polysaccharides, repeating subunits, which is what we see with LPS. Now, there are components that are called mitogens that are essentially T cell independent antigens, so that we can actually use those to see whether or not a lymphocyte is functioning. And we have T cell and B cell specific mitogens. So if we were looking at any particular disease, for example, and we knew that, okay, there's a humoral deficiency, but we do see B cells in the periphery. Now we want to check their function, because maybe they're there, but for some reason they're not functioning. Then we would add the mitogen. The B cells would or would not respond, and that would be a way that we could assess the function of that B cell. So mitogens are used usually in a clinical laboratory. Okay, now what's really important for you to understand as we go into humoral immunity are the antibody isotypes. So all 5 antibody isotypes, and again, there's not much to know about IgD, it really doesn't do a whole lot, but we're going to take a look at not only a little bit about the anatomy, but we're definitely going to dive into the function. And those are things you really, really need to focus on for step 1. These are just some definitions that you're probably already aware of. Whenever we're talking about an idiotype, okay, and maybe you're asked where the idiotype is on the antibody molecule, that's going to be in the variable region or the hypervariable region, because that's where the antigen comes in contact with the antibody, okay? Isotype is a few things. Isotype is similar to the antibody class. For example, IgM is a different isotype than IgG, okay? And IgM has different effector functions than IgG. They can do different things because their constant region is a little bit different and can dictate different effector functions. Now, before we really dive in, there are a couple things I would say you need to know, but really, clinically, you'll never need to know this. But for whatever reason, they love to test it on step 1. So I'm going to give you kind of the quick and easy way to remember this. When we were first trying to define an antibody molecule, okay, we digested the antibody with a few different enzymes. And the first one you see here is papain. So if we expose the antibody molecule to cleavage with papain, what happened is we get a fragmented antibody molecule. So you can see that after cleavage with papain, you wind up with 2 separate binding regions, okay? So there's 1 constant region, 2 separate variable regions, and the big deal here is that in this case, you would not be able to agglutinate, okay, because each part that's left can only bind 1 antigen, okay? Now, that may be tough to remember, but I have a good way for you to remember pepsin cleavage. So you can work backwards. You can remember pepsin and then think papain does the other thing. So for pepsin cleavage, it's going to be a little bit different. Pepsin cleaves below this disulfide bond right here, okay, and is really just going to degrade this constant region, okay? But what you're left with is still a bivalent antibody molecule, okay? So you can still bind 2 antigens. Now, what that means for the antibody and what's left after pepsin cleavage is that you can still agglutinate and you can still precipitate antigens, okay? So that's the most important thing. So how are you going to remember this? You're going to, again, know that pepsin cleaves below that disulfide bond that keeps those 2 FAB fragments together. And we're going to get 2 separate fragments, the FAB prime 2, which is bivalent, and then your constant region. Okay, the constant region gets degraded, and what you're left with is a nice little kind of cup formation. So what you can remember, hopefully, is pepsin, Pepsi Cola. You can have a nice cold glass of Pepsi Cola within that little region because it's a bivalent molecule, and those 2 pieces stay together. All right? That's the best we can do here. Again, clinically, you're never going to need that information, but they like to test it on step 1. So that's why we go through it.